Welcome back, everyone. So I was going through this file for reviewing the cardiovascular system, and I found the blood type that we might need to know for the MCAT. So let's go over this quickly. So as we know, each and every one of us, we have different blood types, and we have antigens. Those are surface proteins on the red blood cells. I believe we have antigens on other types of cells too, but still, these are the main thing about antigens is that they are proteins on the surface of the cell membrane of the cell. There's also the Rh factor. So Rh plus is dominant. This is going into genetics. And an Rh minus person will only create anti-Rh antibodies after exposure to Rh plus blood. So the Rh minus person will only create anti-Rh antibodies. So they make anti-Rh proteins, which are antibodies, after exposure to the Rh plus blood. If they are exposed to the Rh plus blood, the Rh minus person will, now do we say Rh plus or Rh positive? Do we say Rh minus or Rh negative? For blood types, do we say A minus or A negative, or O positive or plus? Let's ask this question quickly since it's important to us. It's urgent right away. O positive is the most common blood type among Americans. About 43% of Americans have an O blood type, of which O positive accounts for about 38% of the population. It is also the most needed blood type because it is most commonly required during blood transfusions. So now we know it's called positive, not plus. So Rh positive is dominant again, and the Rh negative person will produce anti-Rh antibodies if they are exposed to the Rh positive blood. Now we have the blood types here. So the I's, I believe, is like the antigen, and the type of the antigen is on top. So we have IA, IB for AB, and small I for O. So the antigens produced is A for the first one, B is for the second one, A and B for the other one, and no antigens are produced for the O people, O individuals. And we have the antibodies produced, so the A blood type produces anti-B blood antibodies. The B blood type produces anti-A antibodies. AB produces no antibodies, and the O individuals produce both antibodies, both anti-A and anti-B. Now, who can they donate to? So the O people are great donors. They can donate to everyone. They are called the universal donor, and the AB people are called the universal recipients. And it is so if O individuals are universal donors, then they can receive from only O people, because they have antibodies of anti-A and anti-B, so they cannot receive from A or B or AB, right? And for the AB individuals, they can receive from everyone because they have no antibodies. They can receive from AB, A and B, O. And however, they can only donate to AB only, because other blood types have the antibodies that will be against their antigens of the AB people, right? For example, the A blood type, because AB individuals have A and B antigens, they cannot donate to the A person because they have the A person has anti-B antibodies, so it will be against the antigen on the AB, and the same for the B person. How about the O individuals? Still, the O individuals have both antibodies of A and B, anti-A and anti-B, so that's also going to be that's the worst thing for the AB individuals, right? So the first thing you want to do for every blood type is to write down the antigens they have. So we know that people with blood type A have an antigen A, and B people have B, AB people have A, B, and B, and O people have none of them. And then you can write the antibodies. So the antibodies will be the reverse. So for A people, they have antibody anti-B. For B people, they have anti-A. For AB people, they have none of that. And for O people, they have both. And if any of the blood types want to donate to the other ones, they need to it's the interaction between the antibodies from the receiver, right? So to see if the A individual can donate to the B individual, you need to see that the B individual has an anti-A, so the A individual cannot donate to the B individual, right? Can they donate to the AB individual? Because the AB individuals have no antibodies? Yes. So they can donate, the A individuals can donate to AB individuals. As we know, also AB individuals are universal recipients. Cool. We have this thing called the coagulation. So for coagulation, we know that this is about clotting. So coagulation is the blood clotting. So when the endothelial lining of a blood vessel is damaged, the collagen and tissue factor underlying the endothelial cells are exposed. So the collagen and tissue factor underlying the endothelial cells is exposed. This will result in prothrombin turning into thrombin and fibrinogen turning into fibrin, and these are in steps. And then the clots are broken down by plasmin. This is also so clots are broken down by plasmin. What does plasmin do? Is it against coagulation? We also figure out that under the endothelial lining, there's the collagen and tissue factors. Under the endothelial lining are there collagen and tissue factors? What are they? What are tissue factors? And are they outer or inner with respect to endothelial cells? Cool. So we also have here the vasculature. So we know that arteries are thick, they are muscular, they are elastic, they allow for recoil, and they help in propelling blood forward. We have arterioles. We know that the most drop in blood pressure happens here. They are small muscular arteries. And then we have capillaries that are one cell thick endothelial wall, and the diffusion of gases for oxygen and carbon dioxide is easy, and the waste can also transfer here between the capillaries. So the diffusion is easy for NH3, ammonia, and urea, and the same for oxygen and carbon dioxide. There's also the veins, and they have a thin wall. They are inelastic. They may stretch to accommodate lots of blood, but do not have recoil. And the surrounding muscles help pump blood through and contain valves. Something interesting about the veins that I learned in my phlebotomy course is that you witness that the elder people, they have very pronounced veins, and the veins are not stretchable anymore that much, like they used. If you see on the old people's hands or their veins, you can see that it's way too dark. It's black, and it doesn't stretch that much. And so if you try to move the veins with your fingers, you'll see that they don't go back into place, and that's because they have lost their flexibility. Venules are also small veins. So we also have the blood, which is considered a connective tissue. There's erythrocytes. They are formed in the bone marrow. They have no nucleus, mitochondria, or any organelles. They contain hemoglobin to carry oxygen. The hematocrit is the percentage of blood composed of RBCs. And leukocytes, white blood cells, they form the immune system, and they are also formed in bone marrow. And there are two types of leukocytes. There's the granulocytes and the agranulocytes. So the granulocytes are neutrophils, eosinophils, and basophils, and they are non-specific immunity. So they are involved in innate immunity, and they are usually used in inflammatory reactions. Now, agranulocytes, like two examples, which are the lymphocytes and monocytes. So the lymphocytes are for specific immunity. We know, like the B cells and T cells. And monocytes digest foreign matter. They do phagocytosis. They are one of the two types of cells that phagocytose. So if monocytes, so the other type is, I believe, the dendritic cells, or natural killer cells. Not sure. We'll figure that out, but we wrote it somewhere yesterday. Let's see. Yeah, we'll bump into it again. So monocytes digest foreign matter. If monocytes leave bloodstream for organ, they are called macrophages. So if and when the monocytes leave the bloodstream, they are called macrophages. And they're called macrophages because they do phagocytosis. Any cell that does phagocytosis is called a macrophage. And we also have thrombocytes, which are platelets, and they are cell fragments, and they help in coagulation. And they are part of a much bigger cell, which is 1,000 times bigger. And that cell is I forgot the name, but we wrote it somewhere here. Where did I write these? Megakaryocytes. Yeah. So it's the megakaryocytes. Wait, this the thing about phagocytosis happens by macrophages and neutrophils. Oh yes, so we have monocytes as macrophages and neutrophils. So let's write it here too. By macrophages let me refresh this. Okay, so we have that's why we couldn't edit this. Okay, So here we are, monocytes. Cool. So monocytes, if they live bloodstream, they are for organ, they are called the macrophages. So yeah, megakaryocytes lead to thrombocytes too, like the platelets. Now we have two types of pressures for fluid balance. We have the hydrostatic pressure and the osmotic pressure. So if you want to draw these pressures, the hydrostatic pressure moves the fluid out of the blood vessel, and it goes into the interstitial fluid around it. And osmotic pressure is reverse, so it sucks. It is the sucking pressure generated by solids as they draw water into the bloodstream. So this is kind of like what aldosterone does, right? It does sodium reabsorption while the water comes with it as well. So that's why the osmotic pressure doesn't change. The blood osmolarity doesn't change, basically. Now oxygen is carried by hemoglobin, and CO2, some carried by hemoglobin, most exist in the bloodstream as bicarbonate HCO3 minus. And we have a bicarbonate buffer where CO2 plus H2O, CO2 being the gas and H2O being liquid, they give out H2CO3, which is aqueous, and it is the bicarbonate, and that also can dissociate into H plus, which is aqueous, and HCO3 minus, which is aqueous, again as bicarbonate. So if our pH of our blood goes down, we increase our respiration to blow off the CO2, because when our pH is down, it means that we have more H plus, we have more hydrogen in our blood, and our blood has turned more acidic. That happens when we exercise very intensely, and we do increase our respiration in order to blow off the CO2. So when we exercise, the reaction goes to the right, and we have to balance it back to normal by bringing it back to the left side, and that's by respiration, right? Because we want to release the CO2 instead of keeping it, right? Now, if our pH goes up, it means that we have less of the right side, we have less hydrogen, we have more water, and our blood is less acidic, so we do less respiration to trap the CO2. So we don't want to give out too many carbon dioxide as products. We want to keep them as substrates, so we bring it back to normal and we balance it out. And that's our fair share for the cardiovascular system. And we will get back to this soon, but I believe, have a semi-full grasp on the cardiovascular system. And if anything, we can just solidify it with more practice questions on the cardiovascular system. Okay, now we can go to the immune system. Now we need to know that before the cardiovascular system, we had the respiratory system, and I have not gone over this yet, so we can do this later. And we have the hormones. I have studied some of that on my own, but we will go over this in depth as well. And we have stuff about the nervous system, which should be not too major, like it's easy relatively to understand. We have the embryogenesis and the development. That's also something to keep in mind. We have the reproduction, and we have the prokaryotes, the cell, and the eukaryotes. Yeah. But for now, we can continue with, we have the immune system, we have the digestive system, we have the kidneys and the skin, we have the muscular system, and the last one for the biology is the genetics and evolution. And then we have for biochemistry is amino acids. We've gone through this relatively in depth for, but not the second half. We'll go over this again. We have the enzymes. Biochemistry honestly is my arch nemesis, but regardless, we will get a full grasp over this. We have the non-enzymatic protein function and protein analysis. These are our like biotechnologies, and we have the carbohydrate structure and function. This can get ugly at times, still not. We have the lipid structure and function, still should be fine. And we have the DNA and biotechnology, and this is where we have the DNA structure and stuff like that. We have the RNA and the genetic code, which is different than the DNA and biotechnology. And there we talk about the transcription, stuff like that. We have biological membranes, the fluid mosaic model. There's the membrane components. There's the specialized membranes. Ooh, these are the Nernst equation, the Goldman-Hodgkin-Katz voltage equation. Ooh, fancy. We have the mitochondrial membranes. Good stuff. We have the carbohydrate metabolism, section 1, which consists of the glycolysis, glycogen, gluconeogenesis, and the pentose pathways. These can actually get ugly since we need to know the specific enzymes and the substrates and the products for each reaction. And then we have the carbohydrate metabolism. So that was the carbohydrate metabolism, section 1. Again, let's see this as like section 2, which is the aerobic respiration. And then we have the lipid and amino acid metabolism. This can be done in one chapter altogether. We have the bioenergetics and regulation of metabolism. So that's also something that we need to work on. And then we have the behavioral sciences. It's a different story. Like we will probably do the behavioral sciences in a week or two. We need to do practice a lot of those sections before it. We kind of want to do the psych-soc section at the end so that it's fresh in our memory. And we have all these. We have the cognition and the consciousness and language. We have the motivation, emotion, and stress. We have the identity and personality. We have psychological disorders. We have social processes, attitudes, and behavior. We have social interaction. We have social thinking. We have social structure and demographics, and we have the social stratification, which is like the last section, and that's relatively easy. And then we have the physics and math section, which is about kinematics and dynamics. So I personally did a relatively good job on my physics and chemistry, but still I felt that I could improve my chemistry part. So, but about physics, as we can see, is thermodynamics. Here we have fluids. Fluids can be very close and connected to the cardiovascular system. We have electrostatics and magnetism. These can also kind of be intertwined with the biology sections by the authors, by the writers of the MCAT. We have circuits here. Circuits should be relatively easy. We have waves and sound, the Doppler effect, stuff like that. We have the electromagnetism spectrum, light and optics. And we have the atomic and nuclear phenomena. And we have the physics and math. It's this part is like the trigonometry and like how to know the equations and such. We have the reasoning about the design and execution of research. So yeah, we have reasoning that we want to. This might actually be a good thing to study in the first place. So the chapter 11 of physics and math, and then we have database and statistical reasoning. We can get rid of these relatively easily. So we have organic chemistry common names, and we have so the only thing that I didn't cover at the start was the chemistry section. So we can so I'm gonna actually write a kind of milestone based list for tackling every section. So because for example here we can see that we have the high yield sections that we covered, right? But still there is much more depth to the MCAT, as well as the width is not low. So if I want to, so on the top we can write our milestones, right? Milestones. So we have Gen Chem 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Now we have this for every section, right? So after Gen Chem, we have it for the Org Chem. We have it for the biology. Bio. We have it for biochem. Excuse me. Then we have the behavioral sciences. We have the psych-soc. We have psych-soc. We have psych-soc. And then we have sorry, let me stop my recording.